So, welcome back to this lecture, week 4 lecture on Introduction to Professional Scientific Communication. This week we have been discussing about how to write the results section. So, in the previous section we have seen how do you write the results section. So, now we are going to discuss something about the non-textual elements. How do you make tables, how do you make figures that are very effective, which can convey your observation, the best way to present your data. So, let us look into some of these issues. So, these are the tips on effective use of tables and figures in research papers that are sort of listed in this slide here, and these are all from a particular website. The reason I have used this website is that this website also gives you more a detailed discussion on each one of the bullet that I have put in. So, therefore, it would help you because you can go read this particular link, the entire description, therefore, you can better understand. So, the first thing is, first check out what your target journal has to say on this issue. It is very important, when you write a research paper, before you put together all your results, you should know which journal I am writing, whether the journal, what is the guidelines as to the limit, the length of the paper. Some journals are very nice. They will say that they do not put any restriction on the number of pages that you have, but it cannot be 100 pages obviously. So, you easily can, printed pages could be 15, 16, they are fine with that, they do not put, but some journals are extremely stringent. They will say that your entire manuscript should not exceed, for example, 12,000 words, right, which includes figures and everything. They will say that if it is this size, then this many words you calculate and so on. So, the number of printed pages would not exceed, for example, 5 pages. Therefore, now you have to think about as to how you are going to present everything that you found in 5 pages. It is a challenging aspect. So, you need to really decide as to what is the best way to communicate your observations, right, to group the results, to present in different ways. Therefore, you want to do that homework, and before you really arrive at as to how many tables, figures and so on. So, some of the journals limit the number of tables and figures, also have specific guidelines on the design aspects of this display item. They will say that 1 column, 2 column, 1 page is not accepted and so on. So, they will have even, for example, what is the letter that the font you would use in a figure, right, even they will give you these kind of important tips, because that makes the figures and schematics that you make more effective eventually when it gets accepted and published. Therefore, you need to look at that. Next, decide whether the use of tables, figures or text put across key information, right. So, which one I should use, whether I should use the text to convey my results, tables or figures, which way I should be able to put across the information better, that you decide. And after you have decided use on which way you are going to do, choose the display item and best fits your purpose based on what you wish readers to focus on and what you want to present. So, you have to always think that what is the most significant observation. So, how do you highlight that? So, you need to have one or the other way to implement that even when you are making certain figures, right. So, let us look into some of these aspects in this lecture. First, let us look into the tables. So, what are tables, right? The tables, these are to show many and precise numerical values and other specific data in a small space. So, you may have, for example, you are talking about how the temperature fluctuates, for example, in a week, right, 7 days in a particular hilly town, let us say. So, what will you do? So, you will have the day or date, and then you will have the time point, and then you have the exact temperature. So, you can have 3 columns and clearly show, this will mark a small table which conveys everything that you want to say. So, this is basically the precise numerical value that you are applying for the 7 days, right. So, this is one example, right, that you do not need to narrate everything in the text, it is standalone, it can convey. Or to compare and contrast data values or characteristic among related items or items of several shared characteristic variables. So, you may compare one town with another town. For example, the same day same time point how the weather was here as compared to the other city. So now you have two columns, city A, city B, or town A, town B. So you can show exactly how does it fluctuate, right? To show presence or absence of specific characteristic, you can talk about, for example, a remote village and heart of the city, right? You have done the time points, you have measured the temperature. What you are showing is that temperature did not vary between the city and say the rural area, but the particulate matter, the pollution level is very high in the city. So you can bring in a column and show that is very high. The level of that particulate matter in the air is very high in the city as compared to a rural area. So that highlights the difference. So you have a table which can really help you to convey what you wish to display as the major finding of your study. So the best way to decide on the format of the table is to review a number of papers. This is the best thing you want to do. So before you arrive at a decision, you may have certain ideas, but the best thing is you go and read several papers as a reader, and you find which one explains you better, because now you are looking at the data as a reader. You don't know the exact data, but you are trying to understand what others have done. The one that you can understand beautifully is the format that is so helpful to the reader, right? So as a reader, you are able to judge your data better than as the person who sort of generated the data. Now when you go and look at it, probably you will be able to get the best data format that could be the best way you can represent. So after all the data being presented is for the benefit of the reader. Therefore, whatever you find beneficial is the best method or approach you can display your data as well. So therefore, you want to do some homework as to which type of table format that you would like to present. Now we are coming to the figures. The figures are there to sort of again give a summary of the data that you have presented. You may have looked at, for example, I gave you an example of you have looked at the temperature at a certain definite point, like Monday to say Sunday, and twice in a day you measure the temperature in two different cities. Easily you can put in a table, but you have done the same thing for 1 year. So you cannot have a data table which represent every day in a year and 2 data points for every day. Now that would be ridiculous, because nobody can go through that. So what you can do? So you can have a summary of the data. So how do you arrive at the summary of the data? This is where you really make kind of line diagrams, bar diagrams, flow charts, many other such things that would likely to represent the pattern that you have got, the trend that you have got. So to show the trend, patterns, relationship across and between data sets, and the general pattern is more important than exact value. There may be a deviation day to day, but certainly there is a trend, right? So you would expect that around noon the temperature will be much hotter than early morning and for example late evening. That's going to be true whether it's winter or whether it's summer. So you want to prove that, then this is the best way to do that. You give a trend, a pattern using a line diagram and so on. So that is how you arrive at. To summarize research results, for example, again you can present graph, data points, pie chart and so on. To present visual explanation of a sequence of events, procedure, geographical feature or physical characteristics, then you present what is called a schematic diagram. So for example, you have done an experiment. I say that I have injected this drug for 10 days, then I have done a surgery, then I followed it for 15 days. Now I have done various measurements. So now when you are narrating, it becomes very complicated to relate, right? But the whole thing can be the procedure, the protocol as to when did you do the surgery, when was the drug administered, how many days you have allowed the animal to recover. If this can be presented as a schematic, that makes easier for the reader to understand how you are done. So that is one schematic diagram. Or it could be images, like for example, one example I had given was the Western blot, or photographs. You can even have photographs when you talk about, say for example, a deformity. I have a genetic disorder, as a result my facial features changes. So there is no better way than showing a photograph of the face as to, for example, distance between the two eyes, right? Distance between the nose and mouth. So there are differences in certain developmental anomalies. So you have to show the figure, therefore the reader can understand better. So you can show the images, or you can show maps. I gave you an example of how, for example, if you study butterfly, from different localities, different towns and states of the country, where are the places I have isolated the species, I looked at the species, what is the pattern. If I put a map and show it, it makes easy for the reader to understand. So these are the ways you can add the figures. We are going to look into some of those data as to how effectively you can present. So again it is from the Springer website. Figures and tables, what you call as display items, are often the quickest way to communicate large amount of complex information, right? That would be complicated to explain the text. It's very difficult to explain the text, so you better sort of put the data in one or the other form and sort of summarize as to what that particular figure means, what that particular table means. That much easier. I am showing one figure from a research paper. This is a complicated data here, but if I tell you, it will help you to understand quickly even those values that I need not go and detail you. So what is shown here is the one that is shown as exon 1, 2, 3, 4, the line at the bottom, the chromosomal segment of a particular gene. So now you can understand what do you mean by exon 1, 2, 3, 4. If you are a biology student, these are different section of a gene. And then the above there is one CBD, DSPD. This is nothing but a protein which has two domains. This is the protein that is coded by this particular gene. Now these are the two domains. What is shown on the top are the mutations, the individual mutations that are known to be present in many of the patients suffering from a given disease. So these are the different mutations, right? The mutations, because of the mutation, a particular amino acid residue is changed to something else, or the frame is shifted. So each mutation affects the way the protein functions, and the bar above talks about the frequency of the mutation. How many independent families had the same mutation? Now with this information, if you look into, you will find there are what is called as a deletion, large deletions. In exon 2, the occurrence is very high. You can see frequency 14, the bar is like almost like skyscraper. Then you have another mutation which is R241X towards the right side. Again you find a tall bar, which again suggests that these are the mutations that are more frequent in the families as compared to the other mutation. It clearly summarizes. I need not go and say that every mutation what is the frequency. In about 5 sentences I can complete the whole thing, that what is the highlight of the figure. So if I want detail, I can go to the figure and I can get the information. So such kind of figures really help the reader to understand the complex data. So for one to develop this figure would take large amount of time, because you have to go through the literature, every paper, identify which are the families that had the mutation, make a huge table, convert them into frequency table, and then make the figure. It takes time, but that's the effective way of communicating. So you may want to consider such kind of ways to present your data. So this is another data from again another paper which talks about again a complexity in the experiment, right? So there are 5 sets here, identified different color, set 1, 2, 3, 4, 5 and so on, and each one is given a given color, the bar represent each one, and you can see that set 1 has got 2 combination PGM, GFP, the other one have various combinations. So basically here there is an assay that was done to look at the expression of certain genes which are listed at the bottom, ASNS and so on, details in need not bother. So basically you are looking at this fold change, the difference in the expression levels of the gene under various conditions, right? What are the conditions? The conditions are set 1, set 2, set 3, set 4 and set 5. There are various conditions. Now let us not get into that exact specifics of this, what conditions are, but these are 6 different conditions or 5 different conditions. By giving a a color code to each one of them and presenting it in a nice way, now I can easily compare every set with other set, and I can understand as to how each one of the gene expression level varies in various conditions. Now this, if I have to narrate in the text, it is going to take 3 pages, but it can be said in a small bar diagram like thing that is shown. In addition, this bar diagram also talks about the values. The value is that each bar represent the standard deviation of the mean, right? So you have biological replicates, you have experimental replicates, and this is a summary of all these things. And there are stars that are put in there. These stars talk about the significance. You have done a statistical test to see whether the difference that you see is statistically significant, right. So that denotes that yes, indeed it is a significantly different and so on. So this is what it explains when you present the data. Therefore, it is important that you make very good figures that would best explain your data. So you cannot simply, one fine morning you get up and then make it. So you need to think about what is the best way. You have to look at large number of papers as to how people present and come up with the best way that you can present your data. Unless you make your data presentable, easily followed by others, this will not be accepted by the community, because if it does not convey what you wish to convey, the reader is not going to get. If he is not going to get the significance, your findings will not be appreciated, therefore it will not be accepted for publication. Therefore, it is important that you communicate your results in the best way and you find the best method to show your results. Now, I am going to show you another schematic diagram which talks about even the experiment. So, what is shown on the left side, the figure A, is an experimental condition, right. So, what is shown on the top is time in hours, that is from 0 hour to 60 hours. So, that is 60 hours experiment, right. Then you had 8 different groups, right, group 1, group 2, group 3, group 4, group 5 and so on until group 8. Now, each group had different type of combinations. You can see GFP P20 followed by GFP. Let us not get into this details, but what it says is GFP P20 was introduced at 0 hour and the following one GFP was introduced at 36 hours. Now, you have various combinations, right, and then you also say that at 24 hours after introducing the first set, that is GFP P20 in group 1, you have added a chemical which is called as Mg132 for next from 24 to 60 hours the cells basically were maintained in a given chemical. So, that is what it explains now. Now, this is your experimental setup, right, your approach, there are different groups. Now, you are going to talk about the outcome of each experiment with reference to, for example, whether the cell survived or not. For example, percent cell death, that is what is shown in B, and then in C it talks about percent cells with aggregates, certain aggregates that are present in the cell. These are 2 different data points for each one of the 8 groups that are shown here. So, now you can easily relate each bar, which is group 1, group 2, group 3, whatever it is in each of the experiment, whether the percent cell death, that is figure B, or percent cells with aggregate, that is figure C. You can easily compare with A and then appreciate which combination is able to protect the cell or which combination is unable to protect the cell, and whether the aggregates that are forming in the cell have any correlation with the cell death. You can compare all these things, you yourself can compare. So, you can in the textual form you highlight couple of points and leave it, the reader can go and look into that. So, it makes the reader, it makes it very easy for the reader to understand what kind of combination that you have done in terms of experimental setup and how the observations are and what really they mean, possibly the correlations and so on. So, it explains, right. So, these are all schematic, these are all data points converted into bar diagram, these are all statistical analysis, everything come in one place to explain what you have done. So, that should be the approach you should take when you want to explain your complex data with the help of schematics, bar diagrams and so on. I will give you one more example of how a complex data can be presented as a figure. So, again this whatever that you see is from a research paper which talks about small segment of a gene, and that gene, the plan is to delete the gene or delete a part of the gene in the cell now, and then you want to understand as to what happens to the cell if that part of the gene is deleted. This is what is called as knockout animal. When you talk about gene knockout, you basically delete a part of a gene and then look at the effect as to what happens to the cell. So, to delete part of the gene, you need to have understood as to what are the restriction sites, an enzyme that goes and cuts there, where are they and which are the sites that you can use to replace that gene with something else, and if you have introduced that particular construct into the cell, how the gene would be rearranged inside the chromosome and how it can be detected. So, these are the things that are shown here. One on the right side is identified as WT allele, is the wild type allele which has got all the enzyme sites which are identified as the BAMH1, HINT3, the relative position is identified. It identifies where is the particular exon called as DSPD exon, and the orientation is identified by an arrow. And then you talk about how you have generated a construct, so you have replaced the DSPD exon with a neomycin cassette. The rest are all flanking sequences, and then once it goes into the cell and replaces that particular exon, now how that would allele, the knockout allele would look like. So this is how you generate schematics which explains what you have done. Sometimes this is required for you to even understand and design experiments. It's not only for the reader. At times it is required for you to come up with ways to manipulate the gene. So this annotation and making representation of how the genome sequence, the different elements are represented, is very, very important for you to understand as well as convey to others. So this is one more example as to how we can use schematics to better explain. I am going to give you one more example because there are different ways by which people do. So you need to appreciate that. You have to keep that in mind. There is no one rule for everything, right? What is shown here is on the top is a pedigree. Basically, it is a family. It is a 4-generation family that is represented. Here the square is a male and circle is a female. A line that is connecting the two directly is called as the marriage. And then you can see that great grandparents had 2 sons and they are married and they had kids, and then finally in the 4th generation you have 2 individuals that are filled with the black color. These are individuals that are affected with a given disease, right? So it explains everything as to what is the relation, whether it is a male or female, how was the marriage, whether it is affected, unaffected, and whether in fact the one square which is LD916 with a line crossed across represent that he is no longer, he is dead by the time when you do this experiment, right? So that means that he is no longer alive. So even that is explained everything. Then basically what you have done is you looked at the genes by doing an approach called as PCR to look at whether different segments of the genes are present or not, and this particular experiment suggests that there are segments that are absent in these 2 individuals that are having the disease, suggesting that the loss of that particular segment of the gene resulted in the disease, right? That is what explained in this figure. So it's very complex data, but presented in a very simple way. Therefore reader can quickly understand the relationship between various individuals that are there, and then to appreciate the results as to the deletion correlated with the phenotype or the disease status in the individuals that have been assayed here. So again, these are the ways by which you are able to convey complex information, and there is no one single rule that applies to everything. So you have to be innovative even when you create figures as to how best you can explain your data to the reader. Many readers will only look at your display items. You have to understand that, right? So for example, I am working in exactly the same kind of field that you are working in, and you have written a research paper. I don't need to bother about all the introduction because I know exactly what has been done. So what I am interested in? I am interested in what is that something new that you found. So what I do? I don't even wait for reading your results section because I know the background. So I look at your figures, right? I straight go to the figures and try to understand myself what the results mean. Or if I am an editor, right? You submit a paper to the journal that I am handling as editor. It comes to me, then I have to evaluate. So you may have claimed something, your title may say something, your abstract may sell fantastic idea, but I want to see whether whatever you are saying is substantiated, strengthened by the observations and how good your observations are. So what I do? So I go and look at your figures, whether they convey exactly the same thing or does it support your observation or whatever you are claiming that it to be. So the figures would tell me. Now you have to remember that if you have not made your figures in a professional way, they don't look professional, then I would doubt even how good you would have been in carrying out the experiments. But if you have made your figures in a professional way, they look good, they combined well, now then it's going to give me a confidence as to you would have done even your research in a professional way, therefore I would have faith in your results, right? So this is extremely important. So you often you see that when you go to any party, you dress up yourself quite well. The reason is you want to present yourself quite well, right? Otherwise you would have been just like that. You wouldn't have combed your hair, you wouldn't have wore the best possible dress material that you have and so on, but that day you go in a better way. So figures are like that, because you want to present yourself so well on that particular day. Figures are like that, they have to be the best. You may have a data, but unless you process and present the best way, it's not going to convince the authors, the readers, or even the editors. So it's important, right? So therefore, ensure your display items can stand alone from the text and communicate clearly your most significant results. It's extremely important because they have to be standalone. When I look at the figure, I should be able to understand even without reading the results section. So that should communicate on its own. It's extremely important. Display items are also important for attracting reader to your work. So as I mentioned right now, well designed and attractive display items will hold the interest of the reader because they like it. So therefore, they would like to see more and understand what are the different elements that are there. Compel them to take time to understand a figure and can even entice them to read your full manuscript, right? It's like a trailer of a movie again, right? So they will show some snippets of the movie and it will make you curious as to what the movie is about. So that's something like that. You need to make the figures very, very attractive. And finally, high quality display items give your work a professional appearance. This is what I was trying to tell you. Readers will assume that a professional looking manuscript contains good quality science. Thus, readers may be more likely to trust your results and your interpretation of the results, right? So if you go and buy a textbook from a market, if the cover page doesn't look good, doesn't look attractive, if the paper quality that they have used is not really good, the printing quality is not good, though the text may be very good, you may not buy that book because it doesn't look good. So you have not gone through the text. It may be fantastic. It's just that they are unable to print in a good quality material, they are unable to design it properly, but the text is beautiful. But again, the look is something that you looked at and you are not convinced, that you may not buy it. Exactly the same way, your figures, you need to be extremely careful as to that they look like a professionally drawn figure. Therefore, you have to know how to develop figures.